Okay, good morning, everyone. Okay. Morning, everyone. So, thank you so much for joining online. And we have a few participants here in person. So, I would like to introduce Dr. Hardik Pandya. He's Associate Professor in the Department of Electronics and Systems Engineering at Indian Institute of Science, Bangalore. And he's done his PhD from IIT Delhi. And I've been known him from more than 2 years now, a very good friend. And his research is highly cutting edge. I visited him once, in fact, twice now. And once I had a misadventure, but it was a nice place to visit in IC Bangalore. Uh, I will open the floor for Dr. Hardik Pandya to present his talk. So over to you. Thank you so much.
And thank you, Manish, again for the invite. And Very nice to meet my another friend here, whom I met before 6 years, but still he remembers me. So that's a good thing. That's how the collaboration always starts. So let me not waste the time and start with what I'm supposed to do here, right? Give a talk. And I'll be focusing on what we are developing at IASC, which is healthcare technologies. The word advanced because some of the things that I am trying to explore, it's not there because the questions that we are asking are uh, are challenging and also there is no solution to these kind of questions, particularly from the clinical perspective. So as an engineer, we thought about how to find solutions for those kind of questions, and we'll discuss some of these things today. Before I go into the talk in detail, let me quickly, for the audience, tell where I am coming from. So this is, of course, we know our world map, and somewhere here is India, and in India also, it's a big country. Bangalore is somewhere here. This is south part of India, right? So I flew like almost 8,000 kilometers to be here. Very happy to be here. So, having said that, I am right now at IISc, and IISc is one of the oldest university in India. Is also ranked one in the university among all the technology and science universities in India. And it was envisioned is a visionary partnership between industrialist Jamsetji Tata. It was his vision to have a university as good as university at that time in UK, right? So the first vice chancellor and director was a Britisher. So very happy to come to UK and talk and give a talk. So some of the notable directors are on your right side. We have CNR Rao. We have so this is the one. Also, if you see, we have Sir C.V. Raman. Sir Raman spectroscopy that we use, right? He was the one who actually invented it, and a lot of top-notch scientists are the directors or were the directors of IAC. At present, we have a director, Professor Govindan Rangarajan, and like I said, in terms of citations per faculty, it is world's top research university. We are more into research, but at the same time, we also give a lot of importance to teaching. However, compared to IITs, which we call as Indian Institute of Technology, our teaching load is comparatively low. And that's why the research can be better because we can spend more time in research. This is how the institute looks like. It's extremely beautiful institute in the Bangalore, and you can see that the first building that you see here is where the director's office and dean's office is there, and it's very green. You can see it's beautiful walk. It's a gulmohar mark. It's a flowers called gulmohar. Right, you walk and you feel like a prince or princess when you actually walk on this beautiful campus. This is my department, and where I have three laboratories and quickly go through that laboratories as well. So this is the research area that I focus on, where different kind of organs we look into. We look into the brain. We will talk about that. Several implantable devices for Parkinson, for stroke. We're looking at brain tumor delineation. We're looking at liver cirrhosis for non-invasive screening of liver patients just by using their breath. We are looking at the fibrosis in heart by developing a probe. We are looking at the tracheal inflammation by developing the endoscopes and other places, other things. And also more like we are looking at the kidney disorders, particularly acute kidney disorders, by using different kind of biomarkers. We're looking at oral cancer screening. Ear is ENT, particularly in the area of neonatal hearing screening. I'll talk about that, and breast cancer screening and diagnosis. On the left side of the screen, what you can see are some of the sensors. On the right side, you can see some of the surgical tools as well as protein-protein interaction like biosensors, antibiotic susceptibility, and drug delivery tools. So I joined IAC in 2017. Currently, I have 25 members working with me. Very fortunate to have brilliant students who joins us in the institute. And a lot of work is done by them, right? We as a researcher get some time to do research, but most of the time it goes behind guiding the students. So most of the work that I'll represent today is done by the students in my lab. A lot of collaborators, and as we all are supposed to do, publications, patents. I also have a startup. So we are focusing on how to translate the lab research to the market. These are some of the clinical collaborators. Of course, I have academic collaborators, but these are some of the clinical collaborators because these are the people who actually gives us the interesting idea. One is that they don't find the solution for that, and then we see that whether the solution exists in the world, and we are not having in India, or it is not there anywhere. If it's not there anywhere, then we pick up that kind of problem and try to solve it. Right. So we are nephrologists, we have neuropathologists, neurosurgeons, ENT, neurologists, neurophysiology, oncology, and fortunately now we have MD and MS PhD programs. So Dr. Sabri Girishan here, he is a neurosurgeon from Ramaya Hospital, which is next door to IISc, and he is pursuing his PhD. So let's go to the next slide. This is a short video which shows what lab facilities I have so that we can see that if there is anything that we can collaborate or you want to utilize it, please feel free to send me an email. I will be very happy to utilize my facilities and we can collaborate. So the idea of showing you this short video is to show that we have a clean room, which we call as a fab lab. It's class 1000, 10,000 clean room facility. The photolithography area is about class 100. And I have second lab, which is more in the characterization, and one more lab at the animal facility where we do implants. Right. So, and some of the systems that I was able to show it to you. So I'll go through four or five slides, which shows most of the project that is right now we are working on, and we'll talk about some of them today. So we are working on neonatal hearing screening. I'll talk about this today. It's a very interesting problem on how to develop a variable for hearing screening of the newborns. We are working on brain tumor delineation. This is again very interesting problem in the area of brain tumor delineation, particularly how to not resect the tissues which are normal, or how to find the tumor boundary during the surgery. Right. Of course, there is an intraoperative MRI. which is there, but most of the hospitals, including in UK, they don't have intraoperative MRI. And that's why it depends on the expertise of the neurosurgeon how to resect the tissue well. So where is the problem? We will talk about that. We work on the indigenous microcortical arrays for stroke rehabilitation. We're developing first in the world cortical implants for multi-site recording and then acquisition of this part of the healthy brain, encoding it and then decoding it and then encoding back by using a neuromorphic chip. We have a collaborator in IISc, Professor Chitan Singh Thakur. He's developing neuromorphic chips. So once we decode the signals, we can encode it back to the spinal cord. So we are doing that. This is Sabri's research project as well. We are also working on Parkinson rat models, developing the surface implants along with the DBS. DBS already you know is deep brain stimulation electrodes, but how the surface electrodes will enhance the curing in terms of Parkinson. So we work on that. We work on the electronic nose, what we call as a non-invasive technology to measure the breath of a patient, and from the breath can we screen a patient. That's the idea. We do two things. Of course, there is a metal oxide semiconductors, which most of us know, but we now are also moving towards colorimetric sensors, which will show change in color when it's exposed to the breath with certain VOCs in higher level. VOCs are volatile organic compounds. We work on the hydration monitoring system, this particularly for acute kidney disorders, that can show the in real time how much is the body hydrated or dehydrated. This goes to much more than hospitals. This goes in the farms because India, being still 70% of the India is still at the village level and also farming. And a lot of farmers, they work at temperatures which are about 40-45 degrees centigrades, and they don't drink water. They forget to drink or they just don't know when to drink. So if you just give a band, which will show you the time and only a red light, it says, okay, now you drink water. As simple as that. But it will help a lot of patients as well as the normal subjects like farmers. And you can expand it to many other places. We are also looking at how to screen oral cancer. This particular diagnosis is protein-protein interaction when antibody antigen reacts, and we are developing a multiplex platform that can identify the oral cancer from the saliva in just 2 minutes. We are working on cytology scanner. This is a recent NIH grant that we work on with GEHTIC on developing a system which you can see here. This is a schematic of the system where it can screen whether a person has oral cancer or not. Is in oral cancer, when it's a screening, it is not a diagnosis. Screening and diagnosis are different. Screening is, it looks like it may not be. But diagnosis, it is or it is not, right? So we are developing a screening platform. That's why we say cytology. Cytology is study of cells. Histology, study of tissue. Study of tissue is a gold standard, which is diagnosis. We are working on optoelectrical transvaginal probe for cervical cancer. Cervical cancer right now, there is a way to identify cervical cancer is only by looking at the image. But if you can develop electrical plus optical, we combine both the techniques that will help the patient in terms of diagnosis. So we are developing that. We are developing a biomodel probe for breast cancer margin assessment. This is particularly important because, as you know, breast cancer is the second largest cause of cancer-related death in women. And what happens is once the pathologist identifies that yes, the tissue looks suspicious, the surgeon will come into play. When surgeon operates the patient, he or she will remove the tissue and send to the pathologist and suture the patient and send the patient back to the ward or to home. When the pathologist looks at the tissue, 25% of the cases around the world, it's not just India, in UK, in US, everywhere, it comes as a positive margin. What does that mean? That if the tissue is positive, having positive margin, patients still have the cancer right in that particular region. So how to develop a probe that can aid a surgeon during the intraoperative surgery? So that's a challenge. Again, there is no solution except intraoperative MRI. So we're developing one with the help of NIR, which is near infrared rays, and ultrasound. So we are fabricating our own ultrasound transducers using MEMS technique. We are also looking at myocardial fibrosis for the heart tissues to understand whether there is fibrosis or not. Right now, we are doing the ex vivo tissue analysis. The idea is to move towards in vivo. This is another interesting problem. This is called handheld fluorescence imaging system, particularly for the liver transplant. When they transplant the liver, they want to make sure that they don't cut the vessels below it, right? So the system that is right now available costs a lot of money. And can we develop an affordable system, just like how we can see in the schematic handheld system that can show where the vein is? This is by using a marker called ICG, which will have a fluorescence dye and it will have green in color. So can you develop this kind of system? So we're working on that. We have been successful in developing the system, and now we are going to start the testing of the system in patient. We also explore as an intern project vein visualizer. It's fascinating about having the veins, because depending on the BMI, sometimes the vein identification becomes very difficult. And when you draw the blood, if the vein is not identified, it's very painful. So can you do it in real time? Of course, this is a technique which was there somehow. I don't see it commercially available in a lot of hospitals. uh, we need to understand why, but at first thing, whether we can identify the veins or not. So out of my curiosity, I gave as an intern project and we were able to develop this system very well. This few projects are very interesting. See, this one is for on demand drug delivery. Now, this is if you have seen the Abbott patch, which is used for continuous glucose monitoring. But if you want to deliver a drug, for example, for diabetic patient, generally glucomet is a drug that is given, and the patient takes like once, twice, depending on the blood glucose concentration. But suddenly the insulin will spike, and that's what is not good. Can we deliver a drug after every half an hour? So can you develop a microneedle patch like this, and you can put it in your on your hand, and you have an electronic module. So electronic module comes along with the patch. You can throw it and you can put another patch comes with a drug which is loaded, and it's a piezoelectric in nature. Now, all of us know what is piezoelectricity. When you apply a voltage, there is a change in the mechanical, right? So here it will vibrate. So it vibrates at a very low level, not like really vibration that you can see, but enough for delivering the drug. Now you have a mobile. Mobile has an NFC, as everyone knows. So you bring mobile next to that patch, it will deliver a drug. Whenever you want to bring next to it, it will deliver. Right? So across period, like every 2 hours, you give little bit, and that may help. That may, because we have not tested it in patient, but we are developing this thing. This is the first of its kind patch anywhere you will find in the world. There are a lot of microneedles that is there, papers are there, but not like this. So we have filed a patent. Most of the things we have filed a patent, except this vein visualizer. And then we are developing our SEEG electrode. SEEG stands for stereo EEG. Generally, the EEG is, it's a electroencephalogram. Epilepsy is seizures, right? So it's like a fit. Most of the epilepsy can be treated by taking anti-epileptic drugs. But when the drugs are not effective, then that's called intractable epilepsy, where the surgeon has to open the skull, which is called craniotomy, and remove that part of the brain, which is resecting. Before resecting, they insert the electrodes deeper into the brain to find the source localization, where is the source of this epilepsy. That is where the SEEG electrodes, stereo EEG electrodes are used. Okay. Now, in India, we are still importing these electrodes, and these are very costly, not affordable for a lot of patients. So can you develop indigenous SEEG electrodes? That's the whole focus, and we're developing one. Here we are looking at the ECOG. ECOG stands for electrocardiography. When you touch the brain, it becomes ECOG. When it is outside the scalp, it is EEG. So we do the craniotomy in the rat's brain, develop this high density electrode array, and implant it to understand the epilepsy, in which area of the brain is epilepsy. Is generalized epilepsy or focal epilepsy? Is a drug effective or not, to understand the efficacy of the anti-epileptic drugs. So let me start with the first project. These are some of the things that we do currently. So I'm just going to the first project, and I'll be very precise in what we are doing, so that we can learn more things and not going too much into depth. So that the idea is to first understand what we do, and then whatever interesting project is there, we can go into depth through your queries, and I'll try to answer your queries. So neonatal hearing screening, right? This is a problem Not really problem, but this is a screening technique which is around the world. Now, whether it is India, whether it is UK, whenever there is a newborn, they have to go through the screening of the hearing, right? And how it is done? By applying the audio stimulus. When you apply the audio stimulus, you measure the EEG. EEG can be measured with a scalp electrode. Scalp electrode can be placed here. All right. So now just you place one electrode and you measure the EEG. The Now before we go into this EEG measurement, let us understand how it is really helping. Right. You see this video where there are two girls, and you will see the doctor is asking to speak in Kannada. Kannada is a language in Karnataka, which is my state from which I am coming right now in India. So you will see that the doctor is asking, tell your name and speak 1, 2, 3, 4. All right. So the first girl and second girl both will speak, and you see the difference.
ನಿನ್ನ ಹೆಸರು ಏನು? ಒಂದು ಎರಡು ಒಂದು ಎರಡು
ಒಂದು ಎರಡು ಮೂರು ನಾಲ್ಕು ಐದು
ಶಾಲೆಯ ಹೆಸರು
ತೊಟ್ಟ ಟಿವಿ
You can see the difference. The second girl, you may not know the language, but you can see the clarity, right? Both were speaking. First one was saying 1, 2, 3, 4, very difficult to speak. So both were identified with a neonatal hearing screening where they failed. They had a deficit, hearing deficit at the point where they were born. The second girl was intervened at an early stage, and that's why after intervention you can use cochlear implant or there are other techniques to fix it. Most of the time, sometimes it's not possible. But most of the time it is possible. When you can fix at the early stage, there's a lot of difference because your hearing deficit is related to your speech and also related to cognition, right? So this is the very easy thing to understand right from the video. But why we are working on this particular problem? Because 70% of the babies that are born in India as of today, right, they are in villages or primary health centers. And the system to screen those babies, which is the hearing screening, is quite costly, about $20,000. So the test is very cheap, but the system to buy system is very costly. Now, those babies, when they are about 2, 3 years old, then the parents would know they cannot hear. So can you develop a wearable that can help to screen those babies right at primary health center, and that should be utilized by a ASHA worker, is a primary physician, or a semi-skilled personnel, right? You just put a band, press a button, it will show yes or no. For us as a scientist, we want to collect data and understand more. So the technique is called ABR, which is auditory brain response. There is another something called autoacoustic emissions. And I'll tell you what is the technique. That's all the systems that you can see here are extremely right. So first, for every 1,000 babies that are born, about 1 to 5, these are healthy babies have hearing deficit. Two, after every 100 babies that are born in the NICUs, about 2 to 4 are having hearing deficits. So the numbers when you see is huge. Okay. All right. So let us understand how the auditory thing works. So it goes from outer ear to the inner ear to cochlea to auditory thalamus here, somewhere here. And that is what the ABR is used for measurement as a screening technique. ABR, again, I am repeating, it's auditory brain response. So what they see is this peak number 5, 7 peaks are there. The reason of having 7 peaks, there's a reason of having 7 peaks, but mostly peak number 5 is what they observe. If you can see this peaks, that means baby can hear, let the baby go. Again, only one electrode is required, as you can see here, right? That's it. But even for certain babies who are cortically deaf, the ABR will be there, but still they cannot hear, right? So what is cortically deaf? If you further go after auditory thalamus, there is auditory cortex. If you want to measure the signal from here, this is called mismatch negativity, like da, 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 da. There is a standard and deviant. That can you do MMN plus ABR, mismatch negativity plus auditory brain response, using the single band, right? Now, why MMN? Because MMN is the only cognitive event-related potential when the baby is born. Also, when the human baby is born, the skull is not fused completely. There's a current leak, and that's a good thing because we have a better signals during that time. Okay, so what we are trying and what are the existing system you can see here. There is an Indian company called Chauhan. It's really good. only requires like few people to put it together and then use it. What you wanted is easy to use system, right? And the cost also should be really low. So here is a just as an example, this is a cortical response and is a ABR. We are using right now open so right on BCI system, but we are moving to having our own electronic module. I'll I'll play two videos here. The first one is the auditory stimulus generation. Here we see the MMN. See the difference in the auditory stimulus, right? Okay. Now let me play ABR, which is the standard how it is given to the ears, like auditory stimulus. That's how the ABR is measured by giving auditory stimulus, right? Of course, this is a little bit higher decibels. It is a little bit lower decibel for the neonates. So now, can we develop? So we have developed a band. As I told you, we are using open site on BCI board, but we are also having our own electronic module. But this cannot do both the things. So we are now having our own hardware. We are developing it, which will make the system quite portable. This is a band we have tested on few neonates, and we are able to see the another thing is you cannot sedate the neonate. So the way is like when they drink milk from their mother, they sleep, and during that time you can place a band. So band should be such that it will not awake the neonate. It should be soft enough, but should be good enough to acquire the data. So we do a lot of this materials where which material can be used, which electrode we should use, whether you should dry electrodes or wet electrodes, snap electrodes or something else. So now we are also working on making a polymer-based electrode. It's soft enough but good conductivity. So we can add P.PSA, we can add the graphene and other things to try to do that. Okay, so now we have tested with our system versus the reference system. It is working very well. Mismatch negativity also works very well. Also, you can see that the brain gets tired as you go on taking the data and giving auditory signals. So there is some signal processing technique called wavelet. If you do wavelet, then the number of epochs, number of trials would reduce significantly. Right, so we are doing it. We have done for n equals to 3. We have got ethical clearance for n equals to 200. Once we do 200, then we can go to the next system of getting the required trials and the certifications. These are our collaborators, some of the ethical clearance document, some patents and publication. So let me move to the next one. This is on the Parkinson. Now we already know that the Parkinson can be treated by using DBS, which is deep brain stimulation electrodes. The difficulty or the limitation with DBS is sometimes patients have nausea, sometimes vomiting, and we wanted to try that whether as soon as you go deeper into the brain, there will be some damage, whether electrode is a polymer or silicon. Silicon, of course, damage is more than polymer. That's why people are moving towards polymer-based electrodes. However, can we just not go into the brain and just use surface stimulation? Can it be useful for treating epilepsy? We don't know. Sorry, treating Parkinson, we don't know. Or can you combine the surface with DBS and would give us a better effect, right? So that was a question that we asked. We fabricated this device, as you can see here. This is a polymer-based device, a PI, which I say polyamide, and then there are gold electrodes on it. There's a fab technique to do that. And finally, you get this flexible electrode that you place in the rat's brain. In rat's brain, it is not very clearly defined which is the motor area, which is the occipital region, this region. But you can see this somewhere as SMA1, SMA2. Here, red color elliptical, you can see the area. And same thing goes here also. That means that you have to have two electrodes, one on left side, one on right side. And you can apply electrical stimulation to any of this electrode. And you can also acquire the data. To apply electrical stimulation, you have to go for P.PSS because of its higher charge induction capability. But before we implant, we should also know where exactly we are implanting. Are we implanting in the right region or not? Because rat's brain is very tiny. So for doing that, all our right side of the brain controls the left side of the body. Left side of the brain controls the right side of the body. All right. So if I apply electrical stimulation on the right side of the brain, right, this left paw of the rat will move. So you just see, I'll play the video. You concentrate here. I'm applying electrical stimulation, and you see it's moving. That means I'm in the right region. This is a motor area. Right? Again, I'm playing the video, so you can see When there's electrical stimulation, there's a blue LED here, and there's a stimulation here also. You can see here, right? So this is the backpack for the rat with stimulation to understand, and we have further. This is a 1 year old video, right? So we have significantly developed this system now, and we are testing it. This is how you measure whether the rat has Parkinson or not. How to create Parkinson in rat, right? In human beings, there are several different reasons of having Parkinson. In rat, to create Parkinson, we studied a lot of literature and found out that we can do this by in a drug induction of 6-OHDA. That's a drug called 6-OHDA. So you can infuse the drug in the brain and to create Parkinson in 3-4 weeks. But how you know it is Parkinson, right? So the normal rat will be able to hold the rod. This is a rotational rod, so-called rotor rod, for longer time. And the Parkinsonian rat will fall faster. That's one way of looking at it. Second way of looking at it is a tapping test. You will see that Parkinsonian rat will not be able to lift the paw. Right? It's just dragging the paw. Right? These are two different ways of understanding that, okay, the Parkinson has been induced. Once it is induced, then you can open the rat's brain, implant the electrodes, suture it back, let it heal, and you can then take data in real time. Apply electrical stimulation, it will do things better. So here the video is there where it is drinking water. You can see here the cable, so you can acquire the data. We want to move from this wired version to wireless version. So we are now using Inten. Inten is a chip, right? These are all the data that you acquire from control versus PD, and you understand how the power spectral density is different in both the models. We are now developing this, what I said is a wireless system. So you just place in the backpack of the rat and the data is transferred wirelessly. These are 16 channel stimulation. All the channels we can stimulate or all the channels we can acquire, right? So this is something we are trying. We are also making our own DBA system. It's a very different technology. We're trying to have a coaxial kind of cable. So this is where we are in terms of the Parkinsonian things. As I told you, Shabri is not only my PhD student, but also a clinical collaborator, right? So he helps us to do the surgery, and the students, they learn and understand how to perform this surgery. Okay, I'll go to the next one. Again, we stick into the brain only. It's a human brain now. All right, from animal we came to human. Generally, there is a challenge called brain shift. What brain shift is that there is a preoperative MRI. This is a brain for people who have a brain tumor. So when they go for preoperative MRI, the image can be seen in the MRI that, okay, there is the area which is suspicious. The patient is brought to the operation theater where the surgeon will operate the patient. First, they will do the craniotomy. Craniotomy is opening 4-centimeter hole by removing the skull. And when you remove the skull, you find dura. You open the dura, you find brain. Because of the pressure difference, the brain will shift a bit. That's called brain shift. Okay. And because of brain shifting, the image that we see in the preoperative MRI and the image that the surgeon will see, right? Image means actual the brain that surgeon will see where the tumor was, that has shifted a bit. Okay? Now in the you can see that there is an indistinct boundary of tumor. You see like this, very difficult for surgeon. Because of that training and expertise, they are able to resect the tumor completely. Most of the time, except glioblastoma. Glioblastoma is very different kind of tumor. Even one cell is there, it will reoccur. But most of the tumors, they can resect it with accuracy. Where is the difficulty? Because they want to make sure that the resection is 100% successful, they will remove the normal region also. And brain is not like any other organ where you can take extra tissue. Normal region extraction or resection means that there will be some deficits in a patient. So how close you can go to the tumor boundary is a question around the world, right? People have tried biomarkers based, people have tried probe based. The best way is, yes, Manish? Yeah, that's what biomarker based is. So we are what one called label technique. We have to do label free technique because during the surgery, how fast you can do that, right? So here we are trying to develop a probe that can do the ultrasound imaging and can do the electrical impedance tomography. EIT is used for surface and ultrasound can be used for the depth. Right? So combining both the things into a pen like a probe. So that's what our idea is. So being a lab which can do fab, we first understood how the electrical properties of the brain tissues are different. Are they different or not? And we tried to understand that. We developed the entire system that you can see here. Right? We also developed force sensor because we want to measure the mechanical properties. And then we identify that mechanical properties measurement relaxation stiff is better than applying a brutal force. So we'll see that what is relaxation stiff measurement. This is an EIT. This is electrical impedance tomography probe. You can see the chip here, and this is an actual thing a student is working. We will place a water drop on the chip, and you can see the image coming in like 2 seconds. That's it, right? So, and you can save the image, you can collect the data, right? So instead of this, now if you place a tissue, you can understand the same thing. But now you do make this probe so tiny that we can use it in during the 4 centimeter. There is something called QSA system. QSA is used to resect the tissue. Within the QSA is like an endoscope. So you can put, but there is another port where you can insert this kind of pen-like structure, or you can use a robot to hold the pen, because how the pressure is given to the brain for a different clinician would be different, right? So how precisely applying touching the brain is another thing, right? So we are working on continuum robot on understanding whether we can use it or not. But for today, we are not talking about that. So this is an actual ultrasound transducer that you can see with a different kind of, these are the area of it. So what we generally see in ultrasound is a linear or curvilinear. These are 2D, 4 by 4, 8 by 8. And of course, there are linear transducer also. So we have been successful in fabricating this transducer. We use the SOI wafer for a reason that we know that that will stop the etching as soon as it reaches the oxide in DRI, and we always get the same kind of silicon thickness, or the thickness of the diaphragm remains same. You can also use a normal silicon wafer 100, and you can try that double side polished. So moving to another probe again for delineating the healthy from fibrotic heart tissues. Now all of us knows that fibrous is the formation of fibrous connective tissue. Fibrosis is called fibrosis, where the fibrous connective tissues in response to injury is characterized by accumulation of extracellular matrix. And the worldwide fibrotic disease cause over 800,000 deaths. So it's a very important problem to work upon. Can you develop a probe that can be used by a surgeon to identify whether the LHD or RHD, if LHD is affected, can you see that RHD is affected during the surgery, right? It's not like a stent placement. It's an open heart surgery, right? So that's a different thing. Now, but how to do that? So we are using a standard. This is a very standard technique of using the NIR. NIR stands for near infrared rays. You have a source, you have a detector, you send the light, you understand what is the reflection from the heart tissue or the absorbance in it, right? And you collect the data. This human heart tissue data collection is extremely challenging and not easy. From the ethical perspective also, it took a lot of time. We also have registered for the CTRI. That means Clinical Trial Registration of India, which all the data that we take can be used by other groups also. So we have developed this entire probe, and then now we are using Mueller matrices. Mueller matrices will help us understanding the different kind of properties of the light when it is inserted and when the detector connects it. So we have the collimator, linear polarizer that goes to the probe. The light passes through the probe. It is an optical fiber-based probe, so the miniaturization becomes better. And then the light is detected back. It is sent to the linear polarization back to the photodetector. This entire system, again, you can see here, right? We have made it in our lab. This is a system how it looks like. We have right now done study for about 80 subjects, 80 heart tissues, which are patient samples, right? So when the doctor operates a patient, Dr. Prasanna Sima, he will give us the tissue sample. We bring it to the lab and we test it using this particular probe, right? Because it's not easy to go to in vivo before we test on ex vivo. And most of the times we work on the animal models. Very rarely we have get a chance of using the human samples, right? So this is where we have used it, and you can see that various kind of optical parameters you can measure if you go for the Mueller matrix. From that, you can select which one is the best one for understanding this fibrosis. So we did some initial studies. These are some subjects here, results here, and you can see that from healthy and disease RHD, we can see the difference. However, between disease RHD and MVD, there is no difference, right? So and with a large number of patient samples, we will know how and which one actually shows the difference, or there is no difference at all. I'm sorry for the way the representation is done. but you can see that 0.0082 is a significant difference. There are some outliers, some overlapping samples are also there. So we have to further work on it at how exactly we can clearly delineate the tissue samples. Using the same technique, we went to the next one, which is the breast cancer margin assessment. I told you the importance of that, that positive margins are there. So can you develop a probe that can be used by, like I said, the surgeon will remove it, it went to histopathology, and then the pathologist will say that, hey, it's margin positive. So patient has to come back, right? A lot of investment of time, money, and the resources. So can you develop a probe, which I'm showing it to here, which has an ultrasound and NIR, right? NIR, we were able to understand that at 850, 940, 1140, because of the collagen and lipid content of the cancer, right? The way the light works on that particular tissues is very different compared to the normal tissues. Now, can we use that part of the NIR and add the ultrasound to it, right? And integrate it? That becomes a bimodal probe, right? Or multimodal probe. So this we can do it in real time, and that's the whole idea. So here what the probe looks like, right? And then we have done some studies. You can see the probe, and here is a breast tissue. These actual breast tissue samples, fresh biopsy. So we have a collaborator from Assam, which is east part of our of India, and my student goes from Bangalore all the way there in the hospital. They spend like 1 month and collect the data, because fresh tissue samples cannot be shipped so fast or so far, except that your hospital next door. And with that perspective only, IISc now has in 2026, we will have the PG medical school and a hospital on the same campus. So then we don't have to go all the way to Assam. Of course, we love to collaborate still, but you have sample right next door, or not even next door, it's within the campus, right? So this is a fiber-based, optical fiber-based sample, and you can see the data acquisition unit, GUI, a probe. Right now, some of the things we have imported again from United States. Now, what we want to do is, can you replace those components and make it or develop in India? So there is a ceramics laboratory from CSIR. They are developing our fiber optic probes, and we want to utilize those. But before we go to that, you can see that there is a lot of interesting data that is there at different wavelength, and also at for absorbance, reflectance, and detector voltages. So we have done a lot of studies on this. Still, we are doing it. This for n equals to 18 subjects, and we also do the adjacent normal. Sometimes there is a mixed tumor. So how the samples comes from the adjacent normal, cancerous tissue, or mixed tumor samples. This is an ultrasound study. Again, the same thing what I was showing it earlier. Right? There is we are using here aluminum nitride. We are now changing aluminum nitride with zinc oxide. We don't go for PZT because if it is within the body, we don't have to take any chance with PZT since it's not a biocompatible material. However, you have a casing and other things, so we don't have to worry. But most of ultrasound that is used is outside the body to scan. Right? When it goes inside, there should be no question. So that's why we are going for zinc oxide testing. It's a 4-mass process. We are almost ready with the ultrasound transducer, and now the next step is to test it with the Verasonic system, which is a reference system. I go to the next part of my talk, which is on the liver cirrhosis. So we are now developing an electronic nose. Now we all know that how our human nose works. There's an array of sensors which is connected to olfactory lobe, and that olfactory lobe is connected to the neural, right? So we have so many sensors to give us the input, right? We have an optical one. Somebody can talk, so we can hear. For example, if I show you orange and lemon, right? And then you have so many things. You can touch the lemon, you can understand. You can see the lemon, you can understand. You can hear somebody saying this is lemon, you can understand. You can taste the lemon, you can understand. So many sensor inputs goes to the brain. And how now if I ask you not to touch, not to smell, right? Not to hear, not to see, but just to taste, can you still find it out? You can. If I just say, can you just smell it? Can you find it out? Yes, you can. But why you can? Because your brain is trained so many times from the time that somebody taught us, right? Okay, this is how lemon looks like. The lemon tastes like. Taste, nobody can tell until we try. Smell, nobody can tell until we try. But lot of feedback, epochs, number of epochs. That's how AI works, right? Last training data, and your brain works better. That's how your brain works. How can we make an electronic nose which works similar to human brain or human nose, right? So for that, you need to have an array of sensors. That's the first thing and very important thing, because it's an input that goes to the brain. So where we are using it? We are using for fibrosis. This for liver cirrhosis. Liver cirrhosis, as you can see, just before cancer liver cirrhosis falls somewhere here. All right. And when the patient is having liver cirrhosis, we all exhale VOCs. VOC stands for volatile organic compounds like acetone, methanol, ethanol, right? Ammonia. Other than CO2 and humidity, what we are generally taught, right? So now this liver cirrhosis patient would have a higher ammonia concentration in their breath. Can you delineate ammonia particularly from the group of VOCs? And that's very challenging because there can be no sensor which can be sensitive as well as selective. But if you have an array of sensors, then you can have a lot of data, and probably that data you can train it to make it understand that whether we can delineate the patient samples or not from the breath. This is where the one of the sensor that we fabricated is a zinc oxide based sensor. It's very traditional way of fabricating metal oxide based sensors. And this entire system that we fabricated, that's very important because you can use different kind of commercial sensor as well as the fabricated sensors. The difficulty or limitation with the commercial sensor is that it is not sensitive at ppb level. It's parts per billion. Which of the VOCs in our breath is parts per billion. Only when somebody has a liver cirrhosis, then that VOC comes, particularly ammonia comes in parts per million. Right? So commercial sensor can be used for parts per million, but not parts per billion. That means control subject cannot be related with the liver cirrhosis patients. This is how the nanostructure looks like. This is a video that will show you that how we are taking. So we take the breath and in a toddler bag and we connect it and we can get the data in real time. I'll show you the video, play the video for you. Because the idea is that it should be used in the hospitals, right? So it should be easier for the doctor to collect the data or nurse to collect the data and then use the system. We have taken breath sample from healthy controls and from the subjects, and we see this radar plot of averaging. We take for 5 minutes until the sensor shows the response, and we have a lot of data. We do the average and then mean, and to understand that how different patient samples shows what kind of things, healthy controls, patient data, and finally, we see the response of all the sensors together. You can see that there is a array for the ZNO versus different kind of existing commercial sensor like MQ137, MP1901. There is a PID sensor which is extremely sensitive to even ppb level, this PID sensor. But it cannot delineate a particular VOC when there is a group of VOCs in there. So that's a limitation. So can we have a sensitive and selective? That's why all this data is collected. Still, we need to work on it, how to further improve the overall performance of this device. Okay, I'll go to the next one where I talk about the protein-protein interaction. It's a biosensor, right? So this is the idea. The initial idea was to measure the proteins. These are some of the protein. DSG3, it failed because DSG3, we found that it is also in a normal subjects, but ITGFI does PEN and CK19, where the proteins which are present in the lymph node. Now, why we want to measure this protein? Because right now for the oral cancer, when the surgeon operates, they will remove the lymph node and send it to the pathologist to see whether cancer is metastatic or not. That means it's moving from one part to other part of the body or not. And that only happens when it goes to the lymph node. So they remove the lymph node, but lymph node are useful for our immune system, right? So without removing the lymph node, can you just take up a little bit of biopsy of lymph node and identify whether proteins are present? Proteins are present means lymph node is cancerous. If proteins are not present, don't take out the lymph node, right? That's what the idea was. And it will take only like few minutes because sensor is ready with your antibodies just to capture the proteins. So we developed this system. It is an interdigital electrode based system. It's very simple. one-shot IDs if you want to fabricate it. But we went for PCB-based thing because we can now have hundreds and thousands of this one. We can reuse it by putting in oxygen plasma. So all the surface chemistry that you do, antibodies that you load, you put in oxygen plasma, everything will be gone, and you can reuse the chip. That was the idea. We also went for electrochemical sensors, which is also a very traditional way of measuring the proteins. Okay, so this is as simple as that you have a sensing device, this is an impedance analyzer, you connect it, now we have entire system. This is a prototype, old prototype, we have a newer version of it, which is here. So the entire multiplexer system can be used along with this IDs, right? And you just have to connect it and you get the data. This improves the sensitivity by changing the negative electrode, positive electrode width, distance, number of electrodes that changes the sensitivity. We're able to see node positive, node negative samples. We were able to measure it and understand and delineate depending on what kind of protein we are measuring. As I told you that the DSC3 did not give too much of change compared to other proteins. But the same technology can also be used for saliva, right? Because it's still a protein. In saliva, there are different kind of biomarkers. So now this is an ongoing project where we detect the proteins from saliva samples. The same technology can be used for congestive heart failure, right? Because now in congestive heart failure, you have CTN1, BNP, NT-proBNP. These are traditional proteins present in the blood. You cannot use blood directly because of the coagulation. So what you do is you can do the centrifuge, take the plasma and load on the chip, right? And let it incubate at room temperature for half an hour, wash it, and you can see the change in impedance. As simple as that, right? So other clearances, and thanks to the funding agencies, almost wherever we apply over a period of time, initially got a lot of rejection, but then learn how to apply, how to present, right? With all of us, the same thing goes. And over a period of time, things gets better. So now, recently, we have also got international funding from DBT Wellcome Trust, is India Alliance Wellcome Trust, and India Alliance DBT Wellcome Trust, also from NIH, which is a recent project on oral cancer, Pratiksha Trust for all the brain-related activities, and of course, IISc initially to give us a lot of funding to start the lab because we get only the empty space, right? So initially, we require funds so that we can work and bring more funds to the institute. So having said that, thank you very much. You want to know more about the lab, you can scan this. And I'm 7 minutes still left in my talk, so you can ask me any question if you want. Yeah. I may not be able to answer all your questions, so don't worry about it. Yeah, thank you. Absolutely. Yeah, please. I have another yeah, sure. Please go ahead. So yeah. How do we make sure that we use it? Yeah. So what we do is we purge nitrogen after everything, and we wait till the base resistance is achieved. If it is not achieved, sensor is not, we cannot reuse it, right? And we also have a firmware to do the error correction of the base resistance. But most of the time, what we have identified that after you inject the VOC, which is a breath sample, right? It goes to a certain value and then it stops, it saturates, right? And then we stop the data also. Then what we do is we inject the nitrogen and also heat it because we have a micro heater surrounding it. So it will dissolve the gases. Right? So combination of both, we don't use it immediately for the second patient. We have to wait like half an hour to get it to base resistance and then we reuse it. Right? So it is not, and you don't get so many patients in a day, right? It is like one patient in one day or two patients in one day. So a lot of time in between. The only thing is instead of doing the histopathology, which is like punch biopsy, can you use breath? But this can be further extended for diabetes also for people who have ketosis. Ketosis means higher concentration of blood glucose concentration. So in that, the ketone body compounds comes into the VOC. And that's why the people who have a higher glucose concentration, the breath will smell sweeter because of the presence of acetone. So can you identify ketosis by using the same technology? But however, I am not in too much favor with a metal oxide based sensors because it is costly. And also to get this saturation back to normal base resistance. Instead of that, can we use and throw sensors like colorimetric sensors? Right? So that's what we are using now with electrospinning technique. You can just make the entire array. Again, you will require array because of the selectivity. But this array will cost you only like 50 cents. right? So you can use it. It's only changing color. Of course, there's the image processing and the AI, but then you just throw it and put on that. Yeah, thank you. Thank you. Yeah, no worries. Yeah, thank you. Thank you. Yeah, yeah. Hmm. 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 Yeah. So there is a so this particular cases they comes only with a liver problem here, right? It is not for home, right? It is not for general clinic. It's specifically for people who have problem with liver. They want to just see whether liver is cirrhosis or not. So that's how it is there. But the concentration level of ammonia is different for gastric people compared to people who have liver cirrhosis. So that is already in the literature.
Hardik, thank you for the snapshot of portfolio about this
Yeah.
technology. I know the challenges. One
Yeah.
thing is to develop variety of these sensors
Right.
and to take them for the clinical
Mhm.
experience, all active
Yeah.
activity. Of course, we have questions for all of them, but
Yeah,
we
yeah.
go with protein.
Yeah, sure, sure, sure.
What surface chemistry did you
Hmm.
use for selective immobilization of
Yeah.
antibodies or protein?
Yeah.
And did you use any validation technique?
Yes.
To confirm that surface chemistry is working as it.
Yes, yes, yes. We have.
And when we regenerate the surface using oxygen platform, of course, you strip off all the antibodies or whatever immobilization you do. Then you have to actually start over again to immobilize all
Yes.
the surface.
Yes. So we use something called EDCNHS, which is standard surface chemistry utilization for the antibodies. When you use that chemistry, you can have antibodies that will be ready for the ED region, will be ready for supply protein. Is antigen. Right? So that is we cross check that how many antibodies have been mobilized or immobilized to be very precise by using FTIR and XPS. Right. So it gives us a very different curve, right? When the antibodies are present or not. Then when we put the antigen, so this antibodies once we do it, we keep it at refrigerator. Whenever you want to test it, you take it out of the refrigerator, put the proteins with the antigen, either saliva or your plasma, and wait for half an hour. Then you wash it and you measure the impedance. Now that's the first thing. That's how we do the surface chemistry. EDCNHS is one of the chemistry that we are using. We're also trying to understand if we can change the antibodies with heptamer. Heptamer shows much more sensitivity kind of antibodies. Then second thing is how we make sure it is reusable or not. So once we do that, everything goes away. You are absolutely right. But the point is everything goes away. Whether something is left or not, again you can use FTI. It will show the similar like a bare gold because the gold integration. We have to redo the surface, but the electronic waste becomes less. So that is what we are using for the protein. But for me as an engineer to understand how the antigen works, how the antibody works, right? That's why we have a relation. Professor is from Mysore, and we have a student who is expertise is in surface chemistry. So actually working with them, I learned this all the terms that I understand, right? Because surface chemistry itself is it's in itself a very different research area. But I can take another question.
Another thing is about payment devices.
Yes.
Did you use, of course, you have used SOI method. And if you are using it on within screen integration, did you think of using non-SOI substrate for creating materials?
Yeah. So I think I was a little bit fast in the presentation. So I said that if we can go for SOI, when you want to make sure that every time you get the same silicon retina, the diaphragm. But we can also go for double sided silicon, polysilicon, which is like 100 orientation. And if you optimize your RI, then you can get the similar diaphragm. The cost will come down. Here the cost comes down even with SOI because the same. This is what the photo was only for 3 inch, but now we are using 4 inch wafer. So for 4 inch wafer, we can at least accumulate about 60 different transducers, arrays of them, and it's very tiny, like 5 millimeter or 4 millimeter in terms of diameter. And within that 4 millimeter, there are like arrays of transducers. So having a 2D array gives a better understanding than having a linear or curvilinear. So You can do that as long as your DRI is optimized. Now, the deep reactive that we use is in the center phase.
Yeah.
That means from student to student, they keep on changing the parameters. If it is exclusively only for females, you can go ahead with that. Since it is not, so SOI is a better alternative.
Did you think of using surface micromachining instead of going to the surface?
Uh, See, for because you look here at diaphragm. No? So surface micro machining, you have to have a sandwich 2 layers. You will release it and then release that. So you can do that. For example, you create a pit and deposit zinc oxide. Do a CMP. Right on that you grow everything and release by using CMP. Right, but that becomes very fragile. Right, we have used we can use like sacrificial layer zinc oxide. Very easy 1% LCL you can just sacrifice. But what you call it, yield becomes extremely low. And also you need to have the precise orientation of this particular physiologic material. So aluminum nitride is 002, right? If you don't get it, then you don't have physiology.
So what was typical thickness of structure?
1 micron. 1 micron is a PZT or your physiologic material, either using aluminum nitride or zinc oxide. The electrodes on the top and bottom were about 200 nanometers. And diaphragm is depending on what frequency you want. Right, that's the beauty of it. If you want 15 megahertz, or you want to have lower. If you have thicker diaphragm, you can have lower frequency. Thinner diaphragm, you can have higher frequency. What application we are looking is brain or breast cancer. So that we are generally between 12 to 15 megahertz. So depending on that, we have done console simulation and we selected the diaphragm. Oh, yeah. You have question.
So I think we can stop it. So much.
Yeah, no worries. Thank you. Thank you.